about host microbiome interactions under stress with respect to ecology and evolution. There's been an explosion in the last 10 years or so of research on the tiny microorganisms that live on larger hosts. And one thing we've learned from a lot of this work that's pretty clear is that these are largely mutualisms. So both the host and the individual components of the microbiomes are benefiting from these interactions. However, communities communities vary within hosts, and there are some communities that are associated with diseases on hosts. Furthermore, some of those communities even seem to be directly causing the harmful effects to hosts. This is something that we've been calling dysbiosis. And the ultimate cause of this is not immediately clear. It's possible that there's just stochastic factors in community composition and that this community itself is causing it. We can shift it back to a good community just by applying probiotics and rescue our hosts. However, if there's an underlying condition that's causing the formation of these kinds of communities, there's no amount of good probiotics that are going to save us. If there's a condition that causes the microbes that reduce host fitness to have an increase in their own fitness, then this dysbiotic effect will be recreated every time. We need to change the conditions to shift how this fitness correlation works. Oh, it is? Yeah. No, it's not. Can you hear me? In the back. I want confirmation from the back. Okay. Yes. I'm just going to keep going if it misses. That's great. Right, so we have a vested interest in understanding what controls the fitness outcomes here, so where the host fitness is and where the microbiome fitness is, and also not just the outcome, but the correlation structure among the fitnesses. I'll be talking about these questions in the duckweed and its associated microbiome. Duckweed is a tiny floating aquatic plant. It's seen here as just this green schmear in the bottom corner. So I've blown up a few individuals for you so you can see what it is. One individual is just one frond and one root. And then they reproduce primarily clonally, while that individual will give rise to daughter fronds on either side of it, and then those will split off and make more daughter fronds. We know a little bit about the microbes that live on duckweeds ever since the first microscopic lenses. This is a drawing from the 1600s of microbes on a duckweed. And they range from bacteria to fungi to diatoms, among others. If we take these microbes away from duckweed, the duckweed don't die, but they do have reduced growth relative to duckweed where we put the microbes back. Furthermore, oops, keep going the wrong way. When plants have higher fitness, they support more microbes, and when there are more microbes, plants have higher fitness. This is a positive fitness correlation, and these effects are typical of mutualistic interactions. This interaction occurs across a set of variable habitats. Duckweed will live in both pristine natural conditions and also in urban areas highly influenced by runoff from downtown areas. In this system, some relevant questions are whether or not duckweed and microbes are adapting to these urban environments, and whether their shared history of pairs that co-occur might, together with the context that they occur in, alter both the fitness outcomes and those fitness correlations. In terms of urban inputs, you're going to think about zinc, because it's highly important for a number of reasons. It has potential toxic effects on aquatic organisms, and it's really highly present in urban areas relative to rural ones. So a lot of our metal structures contain zinc. It leaches out of this and ends up in water. It's in cars, and it's especially found in tires, which continuously degrade into dust as cars drive on roads. And this dust washes into streams and ponds when it rains. We collected duckweed and associated microbes from around the greater Toronto area, from more urban, closer to the city, within the star, and rural environments. From those sites, we collected individual duckweed, which we then propagated clonally from the lab after stripping it of its normal native microbiome in bleach solution. And we cultured microbes independently of the duckweed in a community format in agar plates. We then recombined these in a full factorial design and ran this experiment including 2 different levels of zinc, both a level that is equivalent to background areas in rural Ontario, as well as a level that's equivalent to how much runoff we'd expect to see in a rain event next to a highway. We grew them in 24-well plates, and here you can see what that looks like. Over the course of the experiment, so from day 0 to day 10, there's been some variable fitness outcomes for the duckweed, and for the microbes, you might see some variation in watercolor here. For the duckweed, we measure fitness not in number of individuals, because that's observer bias, but in pixel area at the end. It's very tightly correlated to the number of fronds, so we think it's a good true measure of the number of individuals. Microbe fitness, on the other hand, is a sample of the water, and is really essentially proportional to that visual clarity. It's just a measure of optical density and how many cells are floating around in there. It's a mixed measure across species, so it's fitness in quotation marks because it's not the fitness of any one species, it's just the total microbial growth. Plants, fitness in the experiment is reduced by that runoff level of zinc, so zinc is a stressor for plants. However, the average microbial fitness was not reduced by that. It's possible that zinc is not a stressor for any of the microbes in our communities. Despite it might not be a stressor on average, but we still might see some patterns with respect to where these things came from. So if we take our average duckweed fitness and arrange it by distance to the city center, we might expect to see that duckweeds from more urban sites have higher fitness in the higher zinc half of the treatment, and that microbes sourced from more urban sites on average have higher fitness when they're from an urban area at high zinc. If we look instead at the low zinc half of the experiment, we'd expect these correlations to disappear. We do see a correlation with distance from the city center for duckweed fitness, as expected. Those urban duckweed are more fit in the high zinc half of the experiment. However, we don't see anything for the microbes, potentially because it's not particularly stressful for them. So there's been no differential selection or community filtering of urban microbes. However, when we look at low zinc, we see the exact same trend for the duckweed. Those urban duckweed are just more fit in general across the experiment. And now we've recovered a trend with microbes. So urban microbes are more fit in general also. And I have some ideas of why that might be, but I'm going to save it and focus instead for now on the fact that that slope between duckweed fitness and distance from the city center has stayed identical. I didn't show the axis, but it has stayed identical, suggesting that there's some predictability in human health effects regardless of the environment. If that's the case, we might expect that not only would duckweed fitness, on average, at low zinc be correlated to the duckweed genotype fitness at high zinc, but it might, if we average a different measure, say the microbe fitness that they support, that might be consistent for the duckweed at low and high zinc as well. Whereas for the microbes, it looks like we're probably going to see a lot more variation across environments and the effect of the different microbes. So for duckweed, we do indeed see these super strong trends. The genotype effect is essentially the same on duckweed fitness or microbe fitness, regardless of the zinc environment. However, for microbe, there's essentially no correlation between the microbe effect in one environment versus the microbe effect in the other. So good microbes at low zinc are not necessarily good at high zinc, whether your perspective is the microbe fitness or the duckweed fitness. So this begs the question whether or not beneficial microbes might rescue unfit duckweed or vice versa. I've been hiding the scale of these plots from you because now I'm going to show it to you, and I'm going to resize them so that they're all on the same scale. And what's going on is there's much more variation in fitness, both duckweed and microbe, across the different duckweed lines than across the different microbial combinations. So a good duckweed can rescue a poorly fit microbial community, but probably not vice versa. So to move on to the next idea, whether or not the shared history of some of these communities and the context in which they found them might interactively influence duckweed and microbe fitness. Our expectation for this is that shared evolutionary or even shared ecological history should shift this plant microbe fitness mean in a positive direction, because selective processes will remove low fitness variance from within species, and there's a positive fitness correlation between the two because it's generally a mutualism. And because if there are species lost that are growing slowly in the microbial community, those will then allow the average microbial community growth in the experiment be higher. So we should see that mean shift up. And so we'd like to compare the sympatric combinations, or duckweed and microbe matched from the same source site, with all those allopatric ones. However, I just told you that there were super strong population level effects, so we can't do that. Instead, what we do is estimate a duckweed effect using that average duckweed, but dropping the sympatric combination, and estimate a microbial effect on duckweed using those microbes, or microbial effect on both duckweed and microbe using those microbe combinations, but again, dropping that sympatric one. Combining the two additively, that's our prediction for where the combination of the microbe and the duckweed should fall. If instead our observed sympatric combinations fall above that, that implies there's some benefit of the shared history, which I'm calling adaptation in quotation marks because it includes multiple processes. Just now change the graph so that this 0 point was our expectation, so that now we can look at all the populations and see if there are, if they're falling in this quadrant where there's benefits to both, cost to both, benefits to one, cost to another, and so forth. And we're also going to link low stress shared benefits to those same things at high stress. So if there are benefits of shared history at low stress, and we then shift things to a totally different novel environment, this high zinc stressor, we might expect some of those benefits are going to be disappearing because they didn't arise in the high stress environment, right? They arose in some lower stress situation, and there may be either sort of genotype by environment interactions that equalize allopatric and sympatric combinations, or potentially even trade-offs in things that were fixed in the low stress environment and the high stress environment. So I'm going to show you all the points. And I've only linked about 5 of them because only 5 of them are different depending on the environment. The rest of them, there's the same effect of shared history in both cases. All of the ones that change, though, do change in the direction of reduced benefits from shared history at higher zinc. Okay. So then we might be interested in what the effect is on the fitness correlations of these two. So I'm going to go back to that same thought experiment. When we remove the low fitness variance, we're not only shifting the mean, we're also removing variation. When we remove variation, then there's less covariation, and there's not much chance that we're going to have a plant microbe fitness correlation happening. If we instead recombine these, we're restoring some of that original variation back, and we can now see that. Another way of saying this is that there might be genotype by genotype interactions that increase the variance in the sample. Same thing goes with a novel context, that zinc stressor. This is the same way of saying that that there might be genotype by environment interactions or species by environment interactions. So how to test this? We go back to our experiment, but for every single treatment combination, we're going to estimate the fitness correlation between plant and microbe. We're going to take those fitness correlations, build them up in a histogram, but then split those histograms apart by treatment, so allopatric versus sympatric and high versus low zinc. We didn't expect to see, we expect to see smaller slopes in this combination, so the sympatric at low zinc, but allopatric combinations or higher zinc should restore variation in slopes. That is indeed what we see, so we see smaller variance in the slopes here for the sympatric combinations at low zinc, but either allopatric combinations or an environmental stressor restores the variation. One interesting thing you can ask about if you're curious is that there's some negative correlation specifically at this low stress allopatric combinations. But just to sum up now, in these plant microbe interactions, we saw that urban environments did structure some fitness context, but some fitness across context, but plants drove most of the fitness variation. We saw that a novel, stressful context reduced the benefits of shared history, and that novel combinations and context restored some of the variation and covariation that was lost in these shared histories. And with that, I'd like to take some questions at this time.